Hello, in this video we will discuss about the starch synthesis and their process. So first of all, you should need to understand about the basic concept about the starch synthesis. So let's begin to understand from the first initial steps for the synthesis of starch. So first of all, we should need to understand the cell structure of the plant. So we are just understanding about the plant cell. So this plant cell is the outer covering is known as the cell wall, and this cell wall is used for the prevention of the lysis of the plant as well as rigidity. So here is the mitochondria and the vacuole. That vacuole will when inflate and more turgid. So in this way the cell wall will prevent to burst the cell. While here is important things that a nucleus is here and the DNA. So here is on the other hand, here you can see this is the chloroplast, and this chloroplast is basically organelle which that is important for the photosynthesis for autotrophic lifestyle. So here is the light when coming, and this is the basically when light will be fall on the chloroplast. So this chloroplast can be moved toward the light is known as cyclosis. So the movement within the cell is known as cyclosis. So here is the outer membrane, and here is the inner thylakoid membrane. This thylakoid membrane, and this is the stroma inside. So the thylakoid membrane when fall the light, so the photosystem 2 to photosystem 1 will transmit the electron via photolysis and hydrolysis of water. So the water will be hydrolyzed and byproduct of oxygen. So the oxygen will be produced. So let's begin to understand. Here is the light reaction. This light reaction is producing the ATP and NADPH. This light reaction is producing ATP and NADPH is used for dark reaction, Calvin cycle. And this Calvin cycle by the fixation of carbon dioxide. After the fixation of the carbon dioxide, it will run and reduction and regeneration will be occur and leave the triose. Triose means that three carbon containing sugar, and this glyceraldehyde we can say that. And this glyceraldehyde basically is the two molecules combined together to form a one molecule of sugar. That sugar is known as the pentose sugar or hexose sugar. So let's begin to understand. First of all, here is the starch synthesis. So the starch synthesis is occur due to the first step is the initially is known as the triose. So the starch synthase enzyme used for to produce the carbohydrates. Carbohydrates means the carbon, hydrogen, and oxygen containing molecules containing food. So for example, grain, wheat, bread, etc., which that is produced from the light reaction in the autotrophic plant, autotrophic organism. So is the autotrophic living organism we will discuss about. What is that? So first of all, here is the prokaryotes. Pro mean before, karyon mean nucleus. So it means the initial animals, initial microorganisms. While here is the algae is the basic initial microorganism or initial plant. While here is the algae produce the 90% oxygen. So that is why throughout the world, so 90% oxygen is come from the algae. So that is why the more photosynthesis and more chloroplast and more storage also. So the blue green algae, so that is why the algae blue green algae is the prokaryotes, and bacteria also contain chlorophyll and volvox spirogyra. This is the algae, and the auto had a higher plant and protozoa. Protozoa is the autotrophic protozoa and heterotrophic protozoa. So here is the chlamydomonas is the flagellated is contain a cup-shaped chloroplast. This chloroplast is moved toward the negative and positive phototactic movement. This negative and positive phototactic movement is used for it. And the euglena is basically here you can see this is the light and move, and it will also contain a bean-like structure is known as the disc-like structure of the chloroplast is used for the light reaction to produce sugar. So here is a heterotrophic protozoa. Heterotrophic protozoa basically is depend to other living organism. So anyhow, I hope you are getting this concept. Next we are discussing now to understand the biosynthesis of starch. So the kinase enzyme for the triose to form a fructose 1,6 bis phosphate is just like a 1,6 bis phosphate. Basically is same structure is like it can move toward the glycolysis. So here I am drawing thus again cell structure, and cell structure contains cytoplasm and cytosol. Basically the cytoplasm contain a cytosol, but the cytoplasm is contain living and non-living matter, while the cytoplasmic matrix is known as the cytosol, which that is major chemical reaction will occur. Why here the plastid, and this plast is known as the amyloplast in this case for in the potato development as well as sweet potato in the buried plant root and tuber formation via a storage of the starch into the amyloplast, that is the plastid. Like a chloroplast is also known as plastid. So let's begin to understand from the triose. So this triose will convert by a 2 triose, a 2 sugar molecule of the 3 carbon containing, which that is come from the dark reaction with the kinases enzyme to produce the fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate is also come from during the glycolysis, again reverse and for the reversion. So in this way, the here is the fructose 1,6 bisphosphate formation to produce the fructose 6 phosphate in the presence of fructose bisphosphatase 1, and this is the water addition, and the phosphate group will be released to form a fructose 6 phosphate. But in the case of ADP used to produce ATP to produce fructose, while on the other hand the phosphoglucose isomerase enzyme used to isomerization of this molecule to form glucose 6 phosphate. So the glucose 1 phosphate formation due to the phosphoglucomutase enzyme. So this phosphoglucomutase enzyme to produce this one. After the phosphoglucomutase enzyme, so in this way the UTP used to release the pyrophosphate, and the addition of one more after the formation of UGP1, so the two UGP 1 molecule form to produce the UDP D-glucose. It means the uridine diphosphate. It means the two phosphate group are present, and this fructose addition here is can be fructose addition to release UDP. It means the UDP will be released via a fructose addition, and the fructose will be bind with the glucose to form sucrose. Is the best example is the sugar cane, which that is the reserve or storage is not starch but the sucrose. So here is the sucrose 6 phosphate formation. So this sucrose 6 phosphate formation is also occur to produce after the releasing of phosphate group by the addition of water to produce sucrose in cytosol. So the cytosol. So remember here is the cytosol. This cytosol is the basically the major mechanism for the synthesis biosynthesis of starch. So here is the sucrose is the UDPs use an invertase enzyme to sucrose remove convert into the fructose separation and UDP glucose formation. So this fructose can be converted into the fructose 6 phosphate and hexokinase enzyme used to ATP use, and phosphoglucose isomerase enzyme used to produce fructose 6 phosphate. And this fructose 6 phosphate is the fructose hexoisomerase enzyme used to produce glucose 6 phosphate. So isomerization will be occur. So remember, but here is we are understanding about the UDP glucose. So UDP convert into the UTP due to the pyrophosphate addition to remove the glucose 1 phosphate, and after this removing of the glucose 1 phosphate, addition of the ATP to remove again pyrophosphate, and this removing of the again pyrophosphate to form a ADP glucose. It means the adenosine diphosphate glucose. It means that two pyrophosphate will be removed, so the two will be left behind. So the two type of molecule can be produced. A UDP glucose and ADP glucose. Both mechanism for the synthesis of starch is same. In the next time we will discuss here. So just remember. So here is the UDP glucose and ADP glucose. Basically the UDP containing glucose and ADP containing glucose will move into the amyloplast, into the plastic. So remember about that. UDP and ADP here is used for the transporter as well as helper, and that is used for the synthesis. So the synthase enzyme, starch synthase enzyme used throughout the mechanism. So in this way here is the ADP glucose will be added with the branch starch branching enzyme 1, 3 and 4. Starch branching enzyme 1, 3 and 4. Every type of enzyme, so branching enzyme, which that is bind with the branch side, carbon number 1, carbon number 3 or carbon number 4. So the 1 to 4 glycosidic bond, 1 to 3 glycosidic bond due to the chain branching. So the ADP will be removed to become again ATP, and on the other hand here is the formation. You can see the addition of the glucose, while on the other hand the starch branching enzyme, so the starch branching enzyme basically is formed, more branching is known as the amylopectin. In this way the breaking down and again repairing and again the binding with the alpha 4 to alpha 6 to 1 glycosidic bond, and alpha 1 to 4 glycosidic bond will be occur for the complex formation is known as amylopectin. So let's on the other hand the granule bound starch synthase enzyme. This granule bound starch synthase enzyme used for the formation of granules. So the granules, a little granule will be formed. This little granule will be stored into the amyloplast, and that will become a more and more again more to grow. So that is why the potato, the smaller potato, basically the same structure of the amylose as compared to the bigger potato. It is not a ripening process or something. So this is the branching. So I hope you are getting this concept. Bye.